Hey guys, welcome to the Circulatory and Cardiovascular System. The vocab is yellow, important info is green, and the headings are purple. You need your highlighters and your annotation pen. Here we go. This is the external view of the heart, the anterior view. Let's look at some of the major anatomy of the heart. First, we're going to start with the auricles. This is the right atrium auricle, and this is the left atrium auricle. Remember that when you're looking at a picture, you've got to imagine that that organ is inside your body. This right here, the section right here, is the right ventricle, and this is the left ventricle. Then you have the major arteries and veins coming in and out of the heart. You have the aorta, the largest artery in the body. You have the pulmonary artery. This is going to go to the left lung, and then this is going to go to the right lung, pick up oxygen for the blood, and then the blood will return back in through the right veins, pulmonary veins, and the left pulmonary veins. For blood to come into the right atrium, it's going to either enter from the top of the body with the superior vena cava, or from the bottom of the body, the inferior vena cava. We're going to look at the internal view of the heart. This is the right atrium and the left atrium. This is the right ventricle and the left ventricle. And then we have the valves that divide the chambers. The first are the atrioventricular valves. That means that they divide the atrium from the ventricle. The one on this side is called the tricuspid because it has three cusps or three flaps. On the left side, you have the bicuspid valve, sometimes called the mitral valve, and it has two cusps dividing the left atrium and the left ventricle. So the way to remember this is that you want to try it before you buy it. All right, the next valves are the ones that are called semilunar valves, and they're called semilunar because they're half moon shaped. There's two right here, and then the other ones are right here. They're named for where they go. So these two in this picture right here actually go up to the pulmonary, through the pulmonary trunk to get to the lungs, and our word for lung is pulmon, so these are the pulmonary semilunar valves. And then these guys right here, they're going up to the aorta, so they're called the aortic semilunar valves. And we'll take a look at the valves more closely in just a second. So where is your heart actually located? If you take a look at this little diagram right here, we know that this is the sternum. This visual is showing ribs 1 through 7, and you can see that this is literally just the heart. The arteries and veins are not shown in this picture. And then you take a look at the human body, and you can see that it is pointing to the left. And one of the little things that I like to note is that when people like to pledge allegiance to the flag, they put their hand over their heart, right? And they put their hand right here, but that's not actually where your heart is. You would really almost need to put it in the middle and have your fingers pointing down. So when you take a look at the internal anatomy, like I just said, these are the atrioventricular valves, the tricuspid and the bicuspid, and then the semilunar valves right here. This one goes up to the lungs, so it's called the pulmonary semilunar valve, and then these guys go up to the aorta, so they're called the aortic semilunar valves. If you're looking from the top down onto the heart, you can see the semilunar valves right here, and then you can see the atrioventricular valves here, and this is a lot easier to see how it's 2 cusps for the bicuspid and 3 cusps for the tricuspid. Layers of the heart wall. Like every body system we have studied, we've got layers for this one too. Here's our prefixes: epi, myo, and endo. Epi means upon or on top, myo means muscle, and endo means within. So the epicardium is the visceral layer of the serous pericardium, so it's right here. Then you have a very thick myocardium. This is the muscle of the heart, and it makes sense that it has to be really thick because your heart is pumping. Muscles are contracting your entire life. And then the endocardium, this is the layer that lines the valves. Now, the cardiac myofibril. This should look very familiar. It looks very similar to the skeletal muscle. We've got a bunch of mitochondria all over the place because obviously here's one too, the heart needs a lot of ATP. You have the sarcolemma. Remember, the action potential runs across the sarcolemma. It goes down the T-tubule to stimulate the sarcoplasmic reticulum to release calcium. Then if you look close up at these lines, remember the sarcomere Z-disc to Z-disc, the M line's down the middle, and then you see the thick and thin filaments in between that are made of the proteins actin, myosin, troponin, tropomyosin. Remember all those? And then the valve function. So this is another view of the valves, the AV valve and the SL valve. Just to note from this slide is that when the AV valves are open, the SL valves are closed, and vice versa. When the SL valves are open, the AV valves are closed. So a heart murmur is actually a problem with whether the valve is closing properly. So it's usually because there's some kind of defect in the heart valve, and what the valve is supposed to do is keep blood from going backwards. And so if the heart valve isn't closing properly, this leads to regurgitation, and it's exactly what it sounds like. The blood is going to leak back up through the valve when it should be closed. So what I want you guys to do is I want you to click on the normal heartbeat versus the heart murmur. And what you're going to notice is that the normal heartbeat is very rhythmic. It's just going to go lubbed up, lubbed up, lubbed up. But when you listen to the murmur, there's going to be an extra beat. It's going to go lubbed up, dup, lubbed up, dup. There's going to be an extra beat, which is what it sounds like, because the blood is actually going back up. So take a second and listen to both, and then move on to the next slide. So what actually causes the heartbeat? All right, so take a look at this picture. We're going to start at the superior vena cava. Blood is going to rush into the right atrium, or we're going to come from the lungs, and the blood is going to rush into the left atrium. As it moves down, you can see the picture right here. That movement of blood actually makes the sound lub that we heard in the previous slide. And then with a really big contraction of both the right and the left ventricle, the blood moves up this way and up this way, and that actually makes the dup sound, going up to the aorta or up to the pulmonary trunk to get to the lungs. So that's what you get, the lubbed up, lubbed up, lubbed up sound. The conduction system of the heart, first of all, our heart cells are considered autorhythmic cells. That means that they fire spontaneously, they act as a pacemaker, and they form the conduction system for the heart. So let's talk about this system. The first part is called the SA node, and it would be great if in the heart there was just a little yellow dot and you could be like, oh, there's the SA node. But in all actuality, it's just a cluster of cells in the wall of the right atrium. And this guy's really important because he starts the whole action potential, the whole heart activity, the excitation. That's the word we use, excitation. Okay, so in the first step, the excitation goes across both ventricles, and then it hits this little pink dot right here. Again, it's not really a pink dot, called the AV node. It's in the atrial septum. So remember, there is a ventricular septum down here and an atrial septum right here. And then the atrial septum, the AV node in the atrial septum, transmits the impulse to the bundle of His. I'm not sure if you guys can see this, but it's like a little dark purple line right here. And once it hits right here, then the action potential, the excitation will spread down the ventricle on both sides along these fibers, and they're called Purkinje fibers. Now, the reason this is colored is to show you where on an EKG you would see this excitation, because that's what it's measuring. And we'll go into this in more detail later. The rhythm of the conduction system, it is obviously very rhythmic, and that's good news for doctors, because if the rhythm is off, they're going to be able to tell usually which area is having the problem. So this SA node fires spontaneously 90 to 100 times per minute. And then the AV node right here fires 40 to 50 times per minute. And if both of those nodes are suppressed, the fibers in the ventricle, these guys, the Purkinje fibers, will still fire 20 to 40 times per minute. So there still will be some kind of action to get the ventricles to contract. Now, if the pace is too slow, then you're going to need an artificial pacemaker to be implanted into your body so that it can keep the rhythm to start the whole excitation process. Now, there's something called ectopic pacemakers, and that's if an extra beat forms at the other site, some other place at a random time. Having too much caffeine or nicotine can make that happen. The timing of the atrial and ventricular excitation. So we know that the SA node is the one who sets the pace because it's the fastest. In 50 milliseconds, it's gonna, that's as long as it takes to spread the excitation across both of the atrium and then hit the AV node. Then there's a 100 millisecond delay at the AV node, and that's because blood has to fill up. And it's also because smaller diameter fibers. It allows the atria to fully contract, filling the ventricles, okay, before the ventricles contract, because you don't want them contracting before they fill up with blood. And then in 50 milliseconds, the excitation will go down the Purkinje fibers and both of the ventricles will contract. So on the EKG, you can see you've got this really big spike right here, which matches the same color as the ventricles. And that's showing the ventricles contracting. Depolarization and repolarization. So we've studied this in the nervous system, and some of the numbers are a little bit different for a cardiac cell versus a neuron. Depolarization, the cardiac resting membrane potential is -90 versus the -70 for a neuron. Excitation spreads through the gap junctions. Show me gap junction. Good job. Fast sodium channels open for rapid depolarization, and then we're going to get a plateau phase. The plateau phase is a 250 millisecond versus a 1 millisecond in a neuron, the plateau phase. And that's when the calcium channel opens. Calcium enters from the outside of the cell, because remember, the sarcoplasmic reticulum is what stores it. Sodium channels are going to close. And then just like in the skeletal muscle, calcium is going to bind to troponin, the alligator head, and then, cause the actin myosin bridge to form tension so that the actual muscle can contract. Then you have repolarization. That's when the calcium channel is going to close and then potassium channels open. And the -90 millivolt is restored as the potassium leaves. So you're going to get back to your resting. Then we have something called the refractory period, and it is very long so that the heart can actually fill up with blood again. And so in your notes, I want you guys to highlight this picture. We're starting at the -70, going up to positive about 20, 30, and then we have our plateau phase. Okay, this is very different from a neuron, the timing. And then you have repolarization, which gets you back to resting membrane potential. So highlight with your two different colors. Okay, the electrocardiogram. I'm sure you guys have all seen an EKG or ECG before, but probably don't know what all those little spikies mean. Okay, so first of all, in EKG, what they're doing is they're measuring the action potentials, okay, that are occurring while your heart is beating. This first little wave right here is called the P wave, and that's when the atrium is depolarizing. Then from P to Q, we call that the P to Q interval. That's the conduction time from the atrium to the ventricle being excited. We call that atrial to ventricular excitation. And then you get this huge spike, QRS. Okay, that's when the ventricle depolarizes, aka the ventricle is contracting. And then you have this a little bit bigger than the P, the T wave, is when the ventricle repolarizes. And then you have your pause, and then it starts all over again. One cardiac cycle, all right, so what that means is that 75 beats per minute, that's the average. One cycle requires 0.8 seconds, and we call that systole and diastole. Systole is when the heart is contracting, and diastole is when the heart is relaxed, okay? And we're talking about both atria plus systole and diastole of both ventricles. Okay, so the end diastolic volume, we call that the EDV. That's the volume of blood, we're talking about blood, in the ventricles at the end of diastole, and the average is about 130 milliliters. And systolic volume, ESV, that's a volume of blood in the ventricle, remember that's the bottom part, at the end of systole, which is about 60 milliliters. So what that tells you guys is that when your heart contracts and the blood is being pumped through, there is never a time that the heart is fully empty. So the stroke volume, the volume of blood ejected per beat from each ventricle, you're just doing the math. 130 minus 60 equals 70 milliliters. Okay, so in those horror movies, when somebody like cuts a jugular vein or something like that, blood shouldn't just seep out while somebody's dying. It's a little bit morbid, but while somebody's dying, the heart is still beating, and so the blood should be coming out in spurts. Okay, and if they were to measure it, it'd be 70 milliliters. Cool, right? All right, this leads us to our blood pressure. And you guys have probably heard like, oh, 120 over 80, and you're like, oh, great, that's awesome, and have actually no idea what that means. Well, we're going to talk about what each of those numbers means and what number you actually really want to have. So blood pressure is a measurement by health professionals, and it's made on the large arteries. Now, to get the best and closest, measure of how your heart is, you'd probably want to do it on your neck, right? That's the closest place that we can get to our heart. However, you guys all know, and you've all had your blood pressure taken, it has to squeeze everything. And since there's other parts there, like your esophagus and trachea, your pharynx, larynx, we're not going to use our necks. And so the next closest place is our arm, and that's why they do blood pressure on your arm. Okay, so the first vocabulary term is systolic. This is the pressure at the peak of your ventricle contracting, okay, aka the contraction of the heart. And this is the top number that you're going to get. The diastolic number, which is on the bottom, is the pressure when your ventricles are relaxed, okay? They relax, so the heart is relaxed. Pressure in the blood vessels decreases as the distance away from the heart increases. So you don't want to do blood pressure necessarily at your ankle because you're not going to get the best reading. Heart sounds, if you have a stethoscope on you, what you would do is you would be listening for the different valves working, okay? If you've ever wondered like why the doctor is moving the stethoscope, I used to think when I was a kid that he couldn't hear my heart. He was trying to find it and couldn't hear it, but that's not true. They actually are listening in different spots to hear the different valves, which is kind of cool. Your pulse, pressure waves of blood. Pressure points are the areas where you can easily palpate. Remember, that's when you use your fingers, and that's for easy monitoring. So you guys can stop for a second and test some of these. I think we've all, tried our radial artery and our carotid artery to take our pulse, but you have other places like at your temporal artery, your facial artery, which is kind of a little bit, up on your mandible. Brachial artery, that's where they put the stethoscope when they're doing blood pressure. You even have a femoral artery, one behind your knee called the popliteal artery. You have one on the side of your leg at the tibial artery, posterior tibial artery, and even one on top of your foot, the dorsalis pedis artery. So what is your resting heart rate? Before you move on to the next slide, go ahead and find your pulse and then set your timer for 1 minute and count the number that you get. It should be somewhere between 60 to 75 beats per minute if you have a normal resting heart rate. Sorry to shock you, but this is a very important artery. This is your femoral artery, and if you guys remember from the bodies exhibit, this artery is super, super big, and that's why it's so dangerous if it were to get severed, okay? So the classification of blood pressure for adults, and that would be 18 years and older, is a normal systolic number is supposed to be below 120. Okay, it used to be that 120 over 80 was the average, but actually now 120 over 80 is pre-hypertensive, and I'll tell you about that in a second. You want your diastolic number, the bottom number, to be below 80. Okay, pre-hypertensive, like I said, is 120 to 139. And then diastolic 80 to 90. Hypertension, when your blood vessels are starting to be blocked. Stage 1 is if your systolic is 140 to 159, and diastolic is 90 to 99. And you guys know you can test these like at Walgreens and CVS. They have even like Target sometimes, Walmart, they have the ones where you can sit down and put your arm in and it'll test you if you have no idea what your blood pressure is. Stage 2, this is getting really dangerous. You're probably going to go to the hospital if you're at stage 2 hypertension. Variations in blood pressure, guys, your blood pressure changes all the time throughout the day, and it also, depending on your stress level, will change as well. So human normal range is variable. Your normal BP, like I said, is less than 120 over less than 80. You can see in this picture right here, this would be a normal passageway through your blood vessel. When you start getting pre-hypertensive, you can see that the artery is becoming thicker. This could be plaque. Hypertensive, like I said, above 140 can be really dangerous if this is chronic. But hypotensive, hypo means below, that's if your blood pressure is too low. And it's not as common. It's usually associated with some kind of illness. But you can see essential arterial hypertension. You can see what happens when the blood vessel starts to fill with plaque. Blood is not going to be able to flow as properly or as easily. So how do health professionals actually test or measure arterial blood pressure? So they're going to put the blood pressure cuff over your brachial artery. They're going to pump the cuff. And what that does is it tightens on your arm. You've felt that before. And it's literally stopping the blood flow. So you can't do this like 20 times on your arm or keep it on there for 10 minutes. Otherwise, blood isn't flowing to your tissue. Then they pump it up and they're looking at this. In this picture, it looks like it's on the wall. A lot of times it's on a little circular dial. It's called a sphygmomanometer. And they're literally watching as the number moves. And as it moves, once the blood starts to come out as they're releasing the air, it'll literally make a little jump. And that first little jump is the first number, the systolic number. That means the blood vessels open enough for the blood to go through. And then it's tick, 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 and the last tick that it makes and it starts to flow normally is the bottom number, the diastolic number. And so what they're measuring is when is the first, after you've closed the blood vessel, when is the first chance or ability for the blood to go through, and that's seen on the sphygmomanometer as a little tick, a little bleep. So if we were in class, we would test ourselves with the blood pressure and the stethoscopes. If you've got one, go ahead and try it. Now, the nervous system is considered the big brother to the cardiovascular system because it controls the heartbeat. Remember, the vagus nerve is very important. If you look at this picture, the vagus nerve does a lot of things. One of them is dealing with heartbeat. The sympathetic nervous system, remember, that's our fight or flight response. That's when we increase our heart rate so that we can either fight or flee. And then the parasympathetic nervous system, remember, deals with relaxing, SLUDD, SLUD, salivation, lacrimation, urination, digestion, defecation, will slow everything back down. Now to the circulatory system. This is a picture of the blood, okay? Actually looking at the microscopic view, you see the red blood cells, you see the white blood cells, and it's literally sitting in a fluid called plasma. There are three main functions of the blood: transportation, protection, and regulation. Just remember that blood is a tissue, okay? It's connective tissue. It just happens to be in liquid form. We get the greatest benefit of our blood is keeping us in homeostasis. It continuously flows through 60,000 miles of blood vessels. If you compare the size of a blood cell to what it's traveling in, it's about 60,000 miles. Okay, first function is transportation. Blood moves through the body, all over the body, and cells can receive nutrients that we picked up from the digestive system, oxygen that we picked up from the lungs, and hormones that were secreted from glands all over the body. And then the cells can dump off into the blood their waste products: carbon dioxide, urea, uric acid, and other secretions. There's a ton of stuff that it can secrete, just basically it's waste. Protection. What does our blood protect us from? Well, it's full of white blood cells, so it protects us from harmful microorganisms and their toxins. And what the white blood cells do is phagocytosis. This is a little picture of a white blood cell sending out its pseudopodia and lassoing this microorganism, and it'll bring it back in and destroy it. We have specialized proteins called antibodies, and we'll talk about those more in detail. And our blood protects us from fluid loss after an injury by clotting. That's what the platelets do. Third function is regulation. Our blood regulates our acid-base balance by using buffers, and buffers neutralize potentially harmful effects of having too much carbon dioxide, too much lactic acid in our blood, especially after we exercise, and other compounds. It also cools us. Our body temperature is regulated by cooling or heating, depending on what we need. If we need to cool down, we sweat. If we need to heat up, our body shivers. And this is all controlled by the hypothalamus. Remember, when in doubt, choose hypothalamus. And our blood also regulates the volume of blood. Okay, so if you are, like, say, in fight or flight and you need to run, then your blood is going to flow to your legs, your leg muscles, so that they get enough oxygen to keep running. The properties of blood that we're going to talk about are color, volume, and pH. First, the color of blood. Oh my gosh, guys, this is so important to me. Your blood is not blue. We've already talked about this, but I'm going to say it again. Your blood is red because it contains the pigment protein hemoglobin. You already know that. Arterial blood is the blood that is full of oxygen and chemically bound to hemoglobin, so it has a crimson red color. It's very dark red. Venous blood, which is often represented as blue in textbooks, is blood that's not as oxygen rich, okay? So it's still dark red, but it has a slightly bluish tint just because it doesn't have as much oxygen. When you see it through your skin, the reason why it looks greeny blue is because it's going through, we've already talked about this, the different pigment proteins in our skin, like carotene and melanin, and that mixture with hemoglobin makes a bluey color. But your blood is red forever and ever. The volume of blood. So I have a picture of these soda bottles here just to give you guys a relation. 8% of your body weight is blood, okay? And most of it is in your vessels, and then the rest is in your heart. It doesn't vary much from day to day, year to year. Males have about 5 to 6 liters, so that's the 3 bottles here. Approximately 8 pints if you like to do it in pints. A female has 4 to 5, so that would be like 2.5. Differences due to average body weight, not actual sex. So if you have a male and a female that are the exact same size, they're going to have the same amount of blood, the same volume. Blood is thicker, denser, and more adhesive than water. You've all probably touched blood. It feels a little bit sticky. That's due to the formed elements, the red blood cells. It causes blood to flow 5 times slower than water. So, like, again, back to the horror movies, when the blood is, sliding down the wall, it shouldn't be going really fast. It should be going slow, okay? That resistance to flow is called viscosity. Blood is therefore considered a viscous substance because it resists water flow more than water. So, if in a horror movie it's going down very fast, it's a very bad horror film. The pH of blood is very specific. First of all, it's slightly alkaline, which means it's a little bit basic. Remember, the pH scale goes from 0, okay, to 14. 0 is the most acidic, 14 is the most basic, 7 is neutral. So the number you guys have to remember, it's very specific. You always want your blood to be between 7.35 and 7.45. The range will stay small despite changes in, like, if you change your diet or cells start to secrete more of this or that, metabolic rate by buffering systems that remove hydrogen ions may be off, but it's still, your pH range is going to still be in this 3.5, 4.5. If the buffers fail, okay, meaning like if your blood becomes too acidic, you get something called acidosis, and that is when your body cells stop working. In this chart right here, acidosis can begin by 7.2. If it gets below 6, you're in a lot of danger. And by 7, if your pH gets to 7, guys, that's a very small range, you could die. Too little acid in the blood is called alkalosis. This isn't very common, but it could happen, especially if you're sick. Possible complications from a change in your pH would be an arrhythmia, meaning your heart rate isn't rhythmic. You could go into a coma, your electrolytes become imbalanced. It's called hypokalemia. So it's very serious if your pH doesn't stay in that range. Okay, now to the white blood cells. So we've talked about red blood cells. They're called erythrocytes. White blood cells, these are the fighters, okay? These are our army inside of our bodies, and they have a lot of jobs. Here we go. First of all, you guys need to know that there's not that many compared to red blood cells. For every 1 white blood cell, we have 1,000 red blood cells. So that's quite a difference in number. Okay, so white blood cells are called leukocytes. They're less than 1% of your total blood volume, and that makes sense because you have so many red blood cells. If you were to count them, there would be 5,000 to 10,000 in a cubic millimeter. Any change in your number, so if you go get your blood tested and your white blood cells are too high, that means that your body is really trying to fight an infection. If your white blood cell count is too low, that means that that infection is winning. Okay, the types of white blood cells, first of all, they all contain a nucleus. Red blood cells actually don't have a nucleus. They're kind of just floating around, picking up oxygen and dropping it off. They're not thinking, they don't know what they're doing, they're just floating in a lazy river. But white blood cells, they are very smart because they have a nucleus, okay? I like this picture right here because this little white blood cell right here, he literally is going out to fight. He is an army ready to fight bad guys. What the white blood cell can do that makes them very special is they can wander outside of the circulatory system, and that's called diapedesis. White blood cells differ in their cytoplasm, their size, and the shape of the nucleus, and that's how you can tell which white blood cell you're looking at. They respond to different staining techniques, so they actually show up different colors under a microscope when you stain them. And there's two groups, and they're divided by their cytoplasm. They're either granulocytes or agranulocytes, and obviously granulocytes mean the cytoplasm is grainy, and then when you put an A in front of something, the agranulocytes mean they do not have the grains, or is my name. Okay, so granulocytes. Cytoplasm contains highly visible pebble-like objects, and those are called the granules. Not these big purple guys, that's actually the nucleus, but all these little grains around it, and remember this is a white blood cell. They're twice the size of red blood cells, and you can see that clearly in the picture, and they contain a nucleus that is split. So you can see this is actually one nucleus, but it's split into lobes. You can see them down here as well. This is one nucleus, even though it looks like two. This looks like it's three, but they're actually just lobes. And they are produced, the granulocytes are produced in the red marrow. Three types: eosinophil, neutrophil, and basophil. The names come from the type of stain that they pick up. So the neutrophil picks up a neutral stain, the eosinophil picks up the eosin stain, and basophil picks up the basic stain. All right, the neutrophils. These are the most abundant. In your notes, go ahead and write down that this is the Osmosis Jones of the white blood cell. So he's like the cop. He's the one first on the scene. He's the one that is going to be super fast. They stain pink in a neutral stain. I know that that doesn't look pink, but it's considered pink. The nucleus is 2 to 5 lobes. This one in the picture happens to be 3. And if you can see right here, they're connected by a bridge. So even though it may look like it's separated, it's all connected. And they make up 60% of all white blood cells. Eosinophils, 1 to 4% of white blood cells in a normal sample will be eosinophils. Their granules stain red in an acid stain known as eosin. And the nucleus has, as you can see here, 2 lobes. Here's a close-up of the eosinophil. Okay, you can see that bridge. I like this picture because it clearly shows the bridge and how it actually is just 1 nucleus, not actually 2. You can see all the granules. These I call the mall cops. They're not very mobile, they're not very active, but once they get there, they can do their job. They phagocytize certain foreign particles, especially produced by allergic reactions, invading parasites, pollen grains, mold spores. So they can still do their job. They're just like a mall cop. They're just a little slow. The basophil, this is the rarest granulocyte, only 0.5% or less of white blood cells in a blood sample. Large granules that stain blue in a basic stain, and their nucleus is often bent into an S shape with 2 lobes. The basophils and mast cells produced a substance called histamine, and you guys have probably heard of antihistamines, right? They cause swelling or inflammation when you have like an allergen that comes in, and it basically is calling the white blood cells to come and help. Swelling tells the other white blood cells where the actual infection is, and what I'd like you to do right now is go ahead and click on the video, and then when you're done, move on to the next slide.
An allergy is an overreaction of the immune system to a normally harmless substance called an allergen. Common allergens include pollen, animal dander, down feathers, mites, chemicals, and a variety of foods. On first exposure, the inhaled allergen enters the mucous membrane lining the nasal passages, where it is taken up by the antigen-presenting cell, which presents it to the T cells. These T cells activate the B cells to release substances called IgE antibodies against the allergen. These IgE antibodies sit on the surface of the mast cells. The mast cells have granules containing chemical mediators like histamine and prostaglandins, etc. On exposure, the allergen binds to the IgE antibodies present on the mast cells, cross-linking them. This results in the release of histamine, prostaglandins, and other mediators into the surrounding tissue. These mediators cause dilation of the surrounding blood vessels and increase their permeability. This results in the nasal stuffiness, sneezing, and mucus discharge of allergic rhinitis. Antihistamines work by blocking the action of histamines at its receptors and thus decreasing the body's reaction to the allergen.
All right, the next category are the agranulocytes. They contain very small amounts of cytoplasmic granules, and there are two types, the monocyte right here and the lymphocyte. Both are produced in red bone marrow and also produced by organs of the lymphatic system. So your lymph nodes, your spleen, and your thalamus also produce these agranulocytes. You can see that the comparison to the red blood cell to the erythrocyte, lymphocytes are very close in size, whereas the monocyte is huge. The monocyte is the largest cell in the blood. It's 3 times larger than a red blood cell and 2 times larger than any of the granulocytes. And one of the ways you can really tell that it's a monocyte is how big the nucleus is, okay? You can see right here the nucleus almost takes up the entire cell. 3 to 8% of white blood cells in a blood sample will be a monocyte. Lymphocytes are the same size as red blood cells. We looked at that earlier. They are the smallest white blood cell that exists. The nucleus is round and large, okay, very similar to the monocyte. It takes up almost all of the cell's volume, and this is more abundant. It's 25 to 33% of white blood cells in a blood sample. Now, what you're going to do on your left page when you get to this section is you're going to do a tree map. And this is how I want you guys to set it up. You're going to put properties on one side and functions on the other. And for a tree map, you put the categories, color, volume, pH, and then transportation, protection, and regulation, and then give me 3 facts about each. The function of white blood cells. So we know that they protect us from disease. They can move out of the vessel called diapedesis. That's what this picture is showing you. Once they're in the intestinal fluid, they can act like amoeba, and they're lassoing out, like we talked about earlier, their pseudopodia, so that they can find infection. They will use the chemicals that are released from other cells and invading microorganisms, and even cells that are being damaged to know where to go. Once they're found, the white blood cell traps the microorganism and engulfs it. That is the term phagocytosis. This video that you guys can watch in just a second shows actually under the microscope a neutrophil, remember the osmosis Joneses, actually doing phagocytosis. It's a pretty cool video. The primary cells used for phagocytosis are these neutrophils and the monocytes. Remember we said neutrophils are really mobile, they're first on the scene. Monocytes are also very active, but they're really big, so they can be used to like phagocytize whole big groups of bacteria, so they're very beneficial. So go ahead and watch the video and then move on to the next slide.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
When more white blood cells arrive at the site of infection, they form a collection of living, dead, broken cells and plasma, and that is what we know as pus. Basically, your pus is like a big war that just happened with some of the army dying and some still being alive, and then all the mass chaos that went with it. But we don't just use phagocytosis to combat disease. What we have are these little guys that look like the shape of Ys. And they are very highly specific proteins produced by our lymphocytes, and those are called antibodies. They act against foreign particles and toxins that enter the body, and the production of these antibodies is what we call our immunity. And so basically what these little Ys do, these antibodies do, is they bind to very specific receptors on the bad guys, and they basically call the white blood cells to come over and attack. So they're like little flags that say, hey, this is the bad guy, you need to catch him. So guys, go ahead and take a second and click on these two links to watch the immune system in action, and then continue with the PowerPoint.
When we pick up a virus, our immune system responds with antibodies that circulate in the bloodstream and stick to the surface of the invading viruses. Scientists had thought that antibodies worked by preventing viruses from getting into healthy cells, but researchers at Cambridge University have shown that this is only a small part of the picture. A team led by Leo James showed that some viruses, including those that cause the common cold, gastroenteritis and the winter vomiting bug, take antibodies with them when they infect cells. Once inside, the antibodies signal to biological machines called proteasomes that live inside the cell, telling them to destroy the virus before it can get to work.
What are white blood cells fighting in 2020? Well, the COVID-19 coronavirus. So please take a second and watch the video about the coronavirus.
Coronaviruses are a family of contagious viruses that can cause respiratory infections from the common cold to SARS. These viruses can mutate rapidly, resulting in the 2019 new coronavirus named SARS coronavirus 2, which has crown-like spikes that characterize the family. As with most viruses, SARS coronavirus 2 commonly spreads from an infected person to others through droplets in the air when coughing and sneezing, through close personal contact, or by touching a contaminated surface and then touching the face. Unless controlled, each infection typically results in 2 to 3 subsequent people being infected. Like other coronaviruses, SARS coronavirus 2 deposits its genetic material into host cells. The viral RNA reaches host elements called ribosomes, which act as factories that produce viral proteins. This facilitates replication of the viral genome and assembly of viral components, producing fully formed replicants that are secreted and further spread infection. Infection caused by SARS coronavirus 2 is known as COVID-19 and can cause fever, coughing and breathing difficulties, along with cold-like symptoms. Serious cases of COVID-19 can cause pneumonia, severe acute respiratory syndrome, kidney failure, and can lead to fatalities. Global health organizations have outlined preventive measures to reduce the chances of SARS coronavirus 2 infection, including refraining from touching the face with unclean hands and frequent hand washing with soapy water. As with previous global outbreaks such as swine flu, COVID-19 has gained global attention and treatment development is being accelerated.
And our final category are the platelets. They are known as thrombocytes. So let's review. Red blood cells are erythrocytes, white blood cells are leukocytes, and then the platelets are the thrombocytes. So if you were going to get a test tube of blood and then put it in a centrifuge, which is a machine that spins and divides the formed elements, you're going to get the majority of the test tube is plasma, and then this little part right here would be the leukocytes, white blood cells, and the thrombocytes, platelets, and then the rest would be your erythrocytes, or red blood cells. So the platelets are formed elements that are fragments of complex cells, and during development in the red bone marrow, they're formed when large precursor cells break apart into little pieces. In small fragments, platelets are released into the bloodstream for circulation. The large fragments are broken down further to form even more platelets. Each platelet contains cytoplasm surrounded by a plasma membrane, cell membrane, no nucleus, but most of the other organelles are found inside the cytoplasm. And if you take a look at the platelets, they are about 1/10 the size of a red blood cell, and their shape can be different, usually round, sometimes oval disc, but this is what you would see in blood. You have the yellow representing the plasma. These purple guys are the white blood cells, red blood cells, and then the platelets. 150,000 to 360,000 platelets would be found in a cubic millimeter in a normal blood sample, and that's less numerous than the red blood cells in that same count. Their job is to prevent fluid loss. They also initiate the formation of blood clots, and if there's ever a break in the blood vessel, then it'll plug it up. So what blood type are you? Let's learn about the ABO and RH blood grouping. Blood grouping is based on reactions between surface proteins, so that's what's sitting on top of the red blood cell, and the special plasma proteins that are floating around in your plasma. All right, the first word you guys need to learn is agglutination. Agglutination, make sure you highlight that. And that is when cells clump together because they're not the correct blood type. So you can see in this these trays here, you guys are going to do a blood typing lab as well. This would be clumped blood or agglutination, whereas this is a normal blood sample. Death occurs when blood agglutinates because the red blood cells get destroyed. If you do know your blood type, you can take a look up here and see the percentages of people who have the blood types. AB negative is the rarest blood type at 1% of the population having AB negative, so maybe you're very rare. And then 34%, 38%, so most people are either O positive or A positive. Okay, an antigen. An antigen is a genetically determined protein, and it sits on the surface of the plasma membrane of a red blood cell. So that's what these are representing. These are all different antigens that are sitting on top of this imaginary blood cell that's not really shown in this picture. The antigen is also scientifically called an agglutinogen. Okay, so we have antigens, and then these Y guys are the antibodies. Let's talk about them. Antibodies are proteins within the plasma. So the antibodies are actually floating around, whereas the antigens are actually on the red blood cell. Antibodies are also called agglutinins, right? These words sound exactly the same, yes? The reaction of an antigen and an antibody determine if blood will agglutinate or not. And I'm going to go through that process and teach you guys how that happens. Now, in this little video right here, it goes really fast, so it's hard for me to talk through it. But this is showing how you have a white blood cell right here, macrophage, and what's happening is antibody is going to be produced because it's having a reaction. So a T cell is stimulating a B cell. The B cells release the antibody. Okay, let's see, it goes too fast. Let's do it again. T cell, B cell releases the antibody. The antibody lands on the bacteria, not really lands, but like locks, okay, docks onto it, and now it's being flagged for destruction. Okay, so when the bacteria comes in, it sends off little chemicals that the macrophage picks up, okay, which is showing that part right there. That stimulates the T cell, which will stimulate the B cell to release the antibodies. The ABO system, there are actually only 2 antigens in the ABO system, and the 2 antigens are A or B. So let's take a look. If you're blood type A, this is what it's showing right here, then you have the A antigen all over your red blood cell. If you're blood type B, you have the B, that's in the yellow, the B antigen all over your red blood cell. Now you can see how they're showing them in different shapes because they're each individual proteins that will only lock with something that fits. If you're blood type AB, you don't have an AB antigen. Instead, you have antigen A and antigen B. And then O, think of O as like 0, okay? O has 0 antigens on the red blood cell. So we call it kind of like a ninja. There's nothing on it. It can't be seen. Hopefully that makes sense. All right, blood transfusion. So back in the day, we've learned from a lot of our blood and guts history of surgery documentaries that we've watched that they tried some crazy stuff back in the day, and one of them was blood transfusions. If blood transfusion is unsuccessful, then the red blood cells die, hemoglobin and bilirubin are released into the body, and then that caused the kidney to fail, and that's how you actually died, was by kidney failure. If you match the wrong blood types, agglutination, do you remember what that means? Agglutination means clumping will occur. So they would just give anybody anybody's blood. They didn't actually know back in the day that these 2 people may not have the same blood type, or if the person lives, they think it's a miracle, but really they just happen to have the same blood type. The RH system. Now this is kind of crazy because this actually is something that I went through myself. So the RH system, if you guys have been wondering, well, okay, I have A blood, but what does A positive mean or A negative? Well, that's where we get into the RH system. It was named after the rhesus monkey, so you can see the RH in rhesus, because that's where it was first discovered. They were doing studies on the rhesus monkey and they found another antigen. And the scientists thought, well, if these rhesus monkeys have this other antigen on their red blood cell, I wonder if humans have it as well because we're also primates. It was later found that the RH antigen is on the red blood cell membrane of humans. So if you have this RH antigen, you are positive, and if you don't have it, you're negative. And that's how you get the positive and negative. Okay, sensitization. So here's a picture of me, and this is my husband, Mike. I am O negative, and he is O positive. All right, so I get pregnant with our first baby. That's Logan right in there. And he is O positive. And if you remember genetics, my husband gave the positive antigen, I gave nothing, so all of my children should always be positive. And because we're both O blood type, there's no other opportunity or option except for them to be O blood type. All right, so here's this little parasite inside my body, and it's positive and I'm negative. So my body freaks out, right, and says, oh, there's a foreign body inside here, it's positive. So, and they start to leak into my blood, and so my body starts to produce antibodies, my little army, to fight this bad guy. But my baby is born, so he's fine. Then I get pregnant with my second child, Liam. And what is inside my body already is the army. So the moment he is born, it takes a couple months, but my body starts to attack, these antibodies start to attack the blood, the red blood cells, and they destroy the red blood cells of my baby. Okay, go to the next slide. So if a mother, like me, has an anti-RH antibody cross the placenta to the second fetus, then the agglutination will occur, and that's called erythroblastosis fetalis. Let's break that word down. Erythro is red blood cell, blast means to destroy, and osis means abnormal condition, fetalis is the fetus, so we don't want to say all that, so the short is hemolytic disease. The child, if they have the situation, will suffer from anemia and hypoxia, which is lack of oxygen, so brain damage occurs if the baby doesn't die. They would have to do a blood transfusion performed before birth, okay, so while in utero, which would provide more red blood cells for oxygen transport. So to solve all of this, if a first-time pregnant woman knows she is Rh negative, she can avoid sensitization, okay, making all those antibodies, by receiving a medical treatment with RhoGAM, and that's what I did. It's a shot that you get, and you have to get it every single time, okay, that you plan on having a baby, because you never know if that baby, if you get pregnant again, that baby's going to do the exact same thing and make you build up the antibodies, which would harm your next baby. Okay, so let's go over the antigens that are found on the red blood cell and then the antibodies that would be floating around in the plasma. So if you're type A, you're going to have the antigen A on your red blood cell. So what would be floating around in your plasma? Well, remember, antibodies are fighting the bad guy. So if you're blood type A, who's the bad guy is B. So floating around in the plasma is the anti-B antibody. And you put the anti in front of it to help you remember. You're against B because you're A. If you're blood type B, you're going to have the antigen. Remember the protein sitting on your blood cell. That's basically the flag that says, hey, I'm B. And then who are you against? What antibody is floating in your plasma? Well, if you're blood type B, you are against A, so you have the anti-A antibody. If you're blood type AB, remember you have two antigens. Remember, there's no such thing as AB antigen. You have antigen A and antigen B. So then who are you against? What antibodies are floating around in your plasma? None, because you're not against anybody because you have both antigens. All right, and now to the ninja. If you're type O blood, there are no antigens on your red blood cell. So who are you against? Well, you're against everybody. So in your plasma, you have both anti-A and anti-B antibodies. So go ahead and use this chart to help you fill in your left page in your notebook. This is including the RH. So if you're positive, that means you have the RH. If you're negative, that means you don't. So this is who you can donate to and who you can receive from. So if you are A positive, you can always, for all of these actually, you can always donate to yourself and you can always receive from yourself. So that's easy, that's the given. So if you're A positive, you can give to yourself, but you can also give to AB, okay, because you share the A, but only if you're positive. Who can blood type A positive receive from? Well, you can receive from yourself and you can receive from negative, because remember, negative means there's actually nothing there, so it would never have a reaction. There wouldn't be any agglutination. You can also receive from our ninja, the O, because remember, there's no antigens, it can't see it. O positive and O negative. Okay, let's do one down here. How about O? O positive can donate to all the positives, okay, because remember, there's no antigen, there's nothing on the red blood cell, so it can sneak into any body. All right, so it can donate to all positives. But who can O positive receive from? Well, it can receive from itself and it can receive from O negative. Why can't it receive from B positive? Well, because remember, who is O against? O is against A and O is against B. And now to the blood vessels. So remember, there always has to be a distinguishing color to show the difference between the arteries and the veins. Books like to use arteries for red and veins for blue, but you could literally use any two colors that you want. It's just showing that they're different types of vessels. So let's take a look. The arterial supply of the brain. So when you watch Blood and Guts, The History of Surgery,
One of the main things that we learned in the video about the brain was that it was so hard to do surgery on the brain because it was so vascularized. There were so many blood vessels going into the brain that it was very hard not to hit a blood vessel and cause somebody to die. All right, next slide. Some of the major arteries are the carotid and the subclavian. So if you're taking a look at this picture, this is the aorta. Remember, it's the largest artery of the body. This is the arch, the aortic arch. And the first one going to the right is the brachiocephalic artery, and this one actually will go down your arm. The one in the middle goes up. This is the left common carotid, and then as it goes up into your head, it's going to branch and then branch again into the left internal and external carotid artery. You've probably heard of the carotid artery. And then the third one to the left is the left subclavian, and subclavian means sub, under the clavicle. All right, so these are the main arteries that supply the head and neck with blood. The carotid is the major supplier and branches into, like I said, the external and internal carotid artery. The right subclavian artery originates from the brachiocephalic, we talked about that one, and the left subclavian artery originates from the aortic arch. They branch into vertebral arteries and thyrocervical arteries, okay? So you can take a look at, here's the thyroacromial artery, this is the mammillary artery, common carotid, the cervical trunk right by the neck, and then they branch, okay? Brachial artery will then branch around and go down your arm, etc. The vertebral arteries pass upward toward the foramina, which are the holes, remember, in your bones of the cervical vertebrae so that they can actually penetrate through the bones. The thyrocervical artery extends a short distance to tissues in the neck, which branch to supply major parts of our neck, the thyroid glands, the parathyroid, the larynx, your voice box, the trachea that goes down to your lungs, our esophagus, which is the tube for food and drinks, the pharynx, which is the throat, and then all the muscles of the head and neck. What this picture is showing right here, this little spot right here, is the subclavian anteriogram. That's showing an aneurysm arising from the thyrocervical trunk. So you can see an aneurysm means that's bulging, the blood vessel is bulging and could rupture, which causes obviously a serious problem. The systemic veins, they're large vessels that are formed by convergence of smaller veins and venules. The systemic veins go towards the heart and arteries go away from the heart. The right atrium is the final destination, and the way that they get in there is either through the superior vena cava or the inferior vena cava. Some veins don't go to either, but instead go towards the liver. We kind of talked about this briefly in the last unit. The hepatic portal vein originates from our digestive tract. The hepatic portal system shuts blood from capillaries, so the really tiny blood vessels of your digestive tract, to the capillaries of the liver, and then they divide into either superior or inferior mesenteric veins, and those are the ones that then enter the liver. And the reason why we do that, remember, is that the liver is going to do a lot with the blood, okay? It's going to clean it, it's going to deal with any of the hormones in there, it, detoxifies the blood, so it's got a lot of functions, and it will help. It'll send the blood from there to the spleen to recycle old red blood cells. The liver receives blood from two sources, the hepatic portal vein and the hepatic artery, and you can see those pictures over here. Blood that is high in oxygen enters the hepatic artery, and blood low in oxygen is going to enter the hepatic portal vein. And remember, guys, hepato means liver. Venous blood, and what we mean by venous blood is blood that came from the digestive organs that's low in oxygen but still has the nutrients absorbed by the intestines, will pass slowly through the capillaries in the liver. The hepatic cells, like I said, remove materials used for metabolic functions. Phagocytic cells eliminate bacteria, etc. that penetrate the digestive lining, and then blood passes through the liver capillaries collected by the small veins that lead into the hepatic veins, and then they're eventually emptied into the inferior vena cava, and then they enter the right atrium, all cleaned up.
So let's see another difference in the human body. It's the circulation to the fetus. So go ahead and play the animation and then click on the next slide. So we can take a look at this again in the animation or in the illustration. Here's the placenta, obviously no baby connected. In the red, you have high oxygenation. This is the umbilical cord. And then the umbilical cord obviously has its own umbilical arteries that connect to the mother's internal iliac artery, internal and external. Actually connects right here. And then it moves up to the aorta, all right? This is the aorta. Remember, it goes all the way behind the body, so this is still the aorta. And then the low in oxygen blood will return up to the, this is the inferior vena cava, into the right atrium and go back through the process to get to the lungs, fill up with oxygen, come back in, and then get back out to the baby, all right? Blood transport routine. So taking blood from the aorta to the tissues and back, the vena cavas, goes in this order. You go from the arteries, which go away from the heart. Arteries get smaller and turn into arterioles. Then they get into the smallest blood vessel, the capillary. Then they're going to go back up to the body through venules, which get bigger and turn into veins, which get you back into the heart. You guys can see that veins are way bigger than arteries. So looking at that in a picture, we go away from the artery. Artery gets a little bit smaller and turns into an arteriole, and then we get a capillary network right here.
It's so small that only one blood cell can fit through at a time, and that's why you have a big network, because if just one was going through at a time, you'd have a major traffic jam. So this is like spaghetti junction. And then it gets a little bit bigger. Now we're at the capillary, I forgot to say, we drop off all of our oxygen, pick up the waste, and then we are ready to go back to the heart. So the capillary gets a little bit bigger. It's called a venule. Then you go up the vein, and you'll notice that veins have valves so that blood doesn't drop back down. And then you get back into the heart to get pumped to the lungs to pick up oxygen, and then go back out the artery and start it all over again. Circle of life. Diseases and disorders. Now, guys, we could do a whole unit on diseases and disorders of the circulatory and cardiovascular system, so I've just picked a few. The first is congestive heart failure, and the causes of CHF are coronary artery disease, hypertension, MI, which stands for myocardial infarction, which is a heart attack, valve disorders, and congenital defects. And there's two different types. Left side heart failure, less effective pump, so more blood remains in the ventricle. The heart becomes overstretched and even more blood remains inside. And so blood backs up and it eventually have nowhere to go. So it's going to go back into the lungs, and that's called pulmonary edema, or the lung swelling. And then suffocation and lack of oxygen to the tissues is the result, and that's how you die. Right side failure is when you get the fluid building up in tissues, and it's called peripheral edema instead of pulmonary edema. Clinical problems, MI, like I said, is myocardial infarction. That's a scientific name for a heart attack. Sometimes it's also called a coronary thrombus, and a thrombus is a blood clot. Death of the area of the heart muscle from lack of oxygen. So if the blood is blocked to this spot or to this spot right here, all of that heart tissue dies. And heart tissue, if you remember, heart cells do not regenerate. They're not like epithelial tissue. And so what happens is fibrosis or scar tissue forms. Result depends on the size and location of the damage. So you can have massive heart attacks and the person dies. Or if it's just a little part, they'll have a minor heart attack and hopefully take care of that and fix it before it becomes worse. Infarction literally means death of muscle, tissue, or organ as a result of the blockage of blood supply to that tissue. And the blockage is usually due to plaque buildup in the arteries because of having high cholesterol or really high saturated fats in your diet. Those are all things that you can take care of. So like I said, here is your thrombus, your blood clot, and if it was right here, this part of your blood vessel is not going to be able to take the blood down to this tissue, and so it will die. And then if it dies, your heart can't pump the blood anymore. More clinical problems. Like I said, a blood clot is called a thrombus. They use clot-dissolving drugs called streptokinase or TPAs and heparin. They can also use balloon angioplasty, and I'll show you a picture of that in just a minute. If a thrombus travels, so if it moves away from the spot, it's called an embolus. Angina pectoris is heart pain from ischemia, and ischemia is restriction of blood supply of the cardiac muscle. So a lot of times people go into the ER because they're having angina. It's severe chest pain or really bad pressure and discomfort. But the heart isn't receiving enough oxygen because the coronary artery is narrowed. All right, bypass surgery. If you watch Blood and Guts like you were supposed to, they talk in detail about how the bypass machine was created, and it was ingenious. So basically what they're going to do is they're going to take a vein from another part of your body, usually the leg, and they will literally, where the section is that is blocked, they are going to sew the vein behind it and then bypass it. Okay, so skip this whole section and then reattach it to another part. So the blood coming up, instead of going this way, will kick over to this side and bypass the blockage. Okay, so sometimes you hear people have triple bypass surgery, quadruple bypass surgery. That means that they've had to do this 2, 3, 4 times in the body. Bypass graft, that's just showing you how they actually graft on the vein. Percutaneous transluminal coronary angioplasty, nobody wants to say that, so they just call it the balloon angioplasty. That is where they put the balloon into the area that has the plaque. They blow it up, all right? And when they blow it up, it literally pushes the plaque against the wall, thins it out. When the balloon is inflated, it stretches the arterial wall, and I like how it says here, it squashes the atherosclerotic plaque. And then after lumen is widened, the balloon is deflated and the catheter is withdrawn, and now you can have blood flow. A stent in an artery, this is like an emergency procedure. This is going to maintain the patency of blood vessels, and you can see the size. This is it closed and this is it open. Here's a stent actually in the carotid artery. So same way, they put the balloon angioplasty through the stent. They put it in, they inflate the balloon, which will open up the stent, and then they remove it. And this is going to be needed if the artery is collapsing and can't stay open. Pacemakers, like we talked about before, remember the SA node is our heart's pacemaker, and if it is not working properly, they have to insert an artificial pacemaker. It's small, it's battery operated, it helps the heart beat on a regular rhythm, okay? And it does that by sending electrical impulses to the heart to help it pump properly. An electrode is placed next to the heart wall, and a small electrical charge travels through the wire. So you can see it goes through here. Most pacemakers are demand pacemakers. They have a sensing device, and it's going to turn the signal off when the heartbeat is above a certain level, and it'll turn the signal back on if the heartbeat is too slow. So it's only going to be used as needed. Demand pacemaker. Internal bleeding. Everybody, this is a picture of massive hematomas or bruises. Everybody gets a hematoma every now and then, but it's literally internal bleeding, hematoma. Varicose veins, we've all heard of these, maybe spider veins you've heard of. Well, as the blood is pumped back to the heart, veins act as one-way valves to prevent the blood from going backwards. So you can see you have these valves here, blood flows up and doesn't go back down. However, if the one-way valve becomes weak, they say, crossing your legs too much, not moving around, sitting too much, some of the blood can leak back into the vein, collect there, and then become congested or clogged. This congestion will cause the vein to abnormally enlarge, and you can literally see it protruding through the skin. The enlarged veins can either be varicose, which means really thick, or they can be spider, and those are usually thinner. They now have laser surgery that will zap the clogged blood and allow it to move through the vein again. So that's kind of cool that we have some new treatments. But the main problem is you gotta be moving. You can't let your blood pool in one area. Cyanosis, this is one we've heard of before. Cyan means blue, osis means abnormal condition. So this is a lack of oxygen in the blood causing the bluish discoloration in the skin and mucous membranes. Most cyanosis is seen as a result of congestive heart disease or pulmonary disease, or as a terminal event in cardiopulmonary arrest. So anytime you see somebody turning blue, this is major. Performing CPR is something that you could do. Obviously, you call 911 immediately. Desirable levels for blood cholesterol for adults. So a lot of these problems, these diseases that we talked about, are due to too much cholesterol in the blood. So let's just remember HDL and LDL. Remember we said H is like heaven and L is like Lucifer, so good cholesterol, bad cholesterol. We talked in the digestive system about how we actually make it. It's a waxy fat or a lipid carried through the blood by lipoproteins. The two main types of lipoproteins are the high density good ones and the low density bad ones. Okay, HDL good cholesterol carries LDL bad cholesterol away from the artery walls, but LDLs stick to the artery walls and can lead to plaque buildup, which is called atherosclerosis. So the total cholesterol should be under 200. LDL should be under 130. HDL should be over 40. And we talked about in the digestive system that the only way to improve your HDL is through exercise. You can lower LDL through diet. Normally, triglycerides are supposed to be in the range of 10 to 190. Among the therapies used to reduce blood cholesterol are your exercise, diet, and if it's genetic that you have high cholesterol, there are certain drugs that can help manage that. Exercise in the heart. So, super important. We talked about in the digestive system that yes, exercise can help you lose weight and stuff like that, but the real reason we want to get aerobic exercise is for our hearts, okay? Sustained exercise increases oxygen demand in the muscles. The benefits of aerobic exercise, and make sure you understand that the definition of aerobic exercise is any activity that works the large body muscles for at least 20 minutes, 3 to 5 times per week. Okay, so you have to get your heart rate up. You can't just walk down the stairs or up the stairs. You have to do it for at least 20 minutes to keep that heart rate up. So the benefits are increased cardiac output, increased HDL, decreasing your triglycerides, improving your lung function, meaning it's easier to breathe, decreasing your blood pressure, and keeping your weight under control. So all very beneficial parts for exercise on your heart. So guys, just say no as much as possible. Yes, you can do this every now and then, but over prolonged periods of time, if this is your diet, you're pretty much preparing yourself for the heart attack that you're going to have later on. Okay guys, I'm going to leave you with a short little video on 5 heart healthy tips so that you guys can live a very long heart healthy life.
9 out of 10 Canadians have at least one risk factor for heart disease, which is our second leading cause of death. But the power of prevention is in your hands, says registered dietitian Daphne Steinberg.
So lifestyle is the cornerstone for prevention and for treatment as well as an adjunct to medication.
She offers her 5 top tips for heart health, starting with food.
Try and keep a healthy diet that's low in fat, low in sodium or salt, and high in fiber. So things like lots of fruits and vegetables to get your fiber, fresh foods to avoid the salt. Try and stay away from fried foods and processed foods as much as possible.
Steinberg says it's critical to read your nutrition facts table and ingredients list closely. A few things to look for: a low sodium food contains 200 milligrams or less per serving. Limit your total daily sodium intake to between 1,200 to 1,500 milligrams, or about half a teaspoon. For trans fats, the labels should read 0, but beware. Some foods may contain trans fat in the ingredient list without showing up on the label, so avoid foods containing partially hydrogenated oils and shortenings. Limit your total fat to no more than 30% of your total calories for the whole day, which includes healthy fats like omega-3s. For fiber, you need 21 grams per day, so reach for fruits and veggies in their solid form, as well as whole grain cereals and breads.
Tip 2 would be to avoid smoking. Absolutely. If you don't smoke, you don't want to start, and if you do smoke, you want to quit.
Smoking is related to about 1 in 5 deaths from heart disease. It can damage blood vessels, decrease the amount of oxygen to the heart, and boost blood pressure.
Tip 3, keep an active lifestyle. So for some people that means, going to the gym on a regular basis. For other people that doesn't really work for them.
That means finding something that does. 30 minutes of moderate activity, meaning your heart rate is elevated, is recommended every day. And that goes hand in hand with her next tip, maintaining a healthy body weight. Steinberg says portion control is key.
We do know that a healthy body weight is important for reducing your risk of heart attack and stroke, and it helps to keep your blood pressure under control.
When it comes to waist circumference, men should not exceed 102 centimeters and women should not exceed 88 centimeters. And finally, reduce your stress.
External stress can put internal stress, like oxidative stress as well. Your body does react to things. Your brain sends out different hormones when you're stressed.
Approaches that have been shown to work include meditation, exercise, and healthy eating. And laughter doesn't hurt either. With Sunnyview, I'm Monica Mattis.